Hi everyone. In the last class of protein metabolism 1, we saw about the digestion, absorption of a protein and amino acids in the body and catabolism of amino acids, the urea cycle and the regulation and disorders of urea cycle. So in this session further we'll study about the metabolism as the objectives include blood urea nitrogen and the clinical significance of it, the principle and the procedure for the determination of blood urea nitrogen and creatinine by the colorimetric and enzymatic methods. So first we'll see the introduction. As urea is a nitrogen compound formed almost in the liver, as we can see the structure of a urea which has a 2 amino group and 1 ketone group. So from the catabolism of amino acids and is the main excretion product of the protein metabolism. The concentration of urea in the serum represents mainly the balance between the formation of urea and the excretion of urea by kidney. So as the urea is the major end product of the protein metabolism in humans formed in liver. The synthesis of urea, it occurs in the 4 stages as transamination, oxidative deamination of glutamate, ammonia, transport and the reactions of urea cycle. So in the last class we studied about the urea cycle. So it's a representation of the urea cycle. And this cycle, any defect in any of the enzyme function of a urea cycle leads to the various metabolic disorders, and the determination of blood urea is very important in many diseases of a kidney. So ammonia is produced in various ways by tissues are rapidly removed from the circulation by the liver and which is converted to the urea. So this is very essential as ammonia is toxic to the central nervous system. So where the urea is a water soluble, the conversion of the ammonia to urea is a kind of detoxification. So it removes the toxic material and converts it into water soluble urea. So this is the importance of urea excretion and its estimation, where the clinical significance of a urea includes causes of increase in serum urea. These are the 3 reasons as pre-renal, renal and post-renal. So under the pre-renal we can see dehydration, pyloric and intestinal obstruction and the chronic intestinal obstruction which leads to the serum urea level. And under the renal, acute glomerulonephritis, malignant hypertension, renal tuberculosis, chronic pyelonephritis, hydronephrosis and congenital cystic kidney. So these are the causes for the increase in serum urea as in the renal part. And as a post-renal, the enlargement of a prostate, stones in urinary tract, structure of the urethra and the tumors of the bladder affecting the urine. So here the any cause of obstruction to urine outflow will be the causes for the increase in serum urea level. And the causes of decreased urea as cirrhosis of liver, acute yellow atrophy of liver, severe acidosis and nephritis and also pregnancy and the low protein diet and severe liver diseases which leads to the reduced plasma urea concentration. So next we'll come to the estimation of urea, where the blood urea, it's estimated by chemical method as a DAM, that is diacetyl monoxy method, and by enzymatic method also there. First we'll see the chemical method. When urea is heated with diacetyl containing 2 adjacent carbonyl group under acidic condition, yellow colored compound is formed. So the OD of the color developed can be read at the 420 nanometers. So here the intensity of the color depends on the concentration of the urea in the serum. So this includes the principle. And as a in procedure column we can see the blank standard and test. We'll take it as 3 tubes and will include protein free filtrate and the working standard distilled water, DAM reagent and acid mixture for the test tube as given in the column. So after this the color developed will be read at 420 nanometer. So the concentration of a urea will be proportional to the color of the tube. So the calculation part includes as the concentration of urea in 100 ml of blood is equal to the OD of test minus OD of blank divided by OD of standard minus OD of blank is multiplied with the concentration of a standard by volume of a test, how much we have taken into 100. So finally we'll get the concentration of a urea in the given blood will be measured in mg per dl. So blood urea nitrogen is measured is equal to blood urea level into 28 by 60, that's a normal value will be 5 to 20 mg per dl. So by the enzymatic method we'll be using as urease GLDH method. The principle includes enzyme urease hydrolyzed urea in the given sample to form ammonia and carbon dioxide. So when the enzyme urease gets hydrolyzed in the given sample with the urea, it forms a ammonia and carbon dioxide. So there ammonia formed further combines with the oxaloglutamate or the ketoglutarate and NADH2 to form a glutarate and NAD. So the rate of oxidant of NADH2 to NADA is measured as decrease to concentration which is measured at 340 nanometer. So the equation will be urea with the 2 water molecule in presence of urease enzyme, it forms ammonia plus carbon dioxide. So the overall reaction will be represented as ammonium reacts with the oxaloglutarate, it forms a L-glutamate with the NAD plus water in presence of GLDH enzyme. So here in the column we can see the blank standard and the test. As it is given 10 microliter will be added to the each test tube as a distilled water and urea standard will be added to the standard. The sample for the test and the concentration of urea will be 50 microliter and the OD will be measured at 340 nanometer. So the calculation includes as OD of a test minus OD of a blank divided by OD of standard minus OD of blank into the concentration of standard by volume of test into 100. So it's measured in terms of mg per dl. So next we'll see the blood urea nitrogen, that is we can call it as BUN. So sometimes it's used as a measurement of serum urea. So the half of the molecular weight of urea is nitrogen because the urea structure itself we can see the amino group is present, so the nitrogen part of it. So half of the molecular weight of a urea will be nitrogen. So the normal range will be serum urea is 20 to 40 mg per dl, the BUN 5 to 25 mg per dl. So the abnormal conditions where it leads to the high serum urea will be tissue protein catabolism, it increased, means negative nitrogen balance it's there, and this occurs in fevers, wasting disease, thyrotoxicosis, diabetic coma after trauma or a major operation. So in all these conditions the high serum urea level will be there. And in abnormal conditions, as excess breakdown of blood protein also leads to the increase in urea level, as leukemia, release of leukocytes protein, and the GIT bleeding, hemoglobin and plasma protein can be released into the gut. So thus there will be a increase in the blood urea. And the creatine and creatinine, here we can see creatine constitutes about 0.5% of total muscle weight. It's synthesized from the 3 amino acids as glycine, arginine and methionine as guanidinoacetic acid to the creatine, and creatinine phosphate formed in the liver. So here creatinine is present in muscle, brain and liver. Creatine kinase. Here we can see the conversion of creatine to creatinine and the phosphocreatinine by the creatine kinase enzyme. So the clinical significance of a creatine and creatinine will be stored creatine phosphate, which is high energy compound stored in the muscle, serve as an immediate store of energy. Interconnection of these phosphocreatine and creatine is an indicator of metabolic process of muscle contraction. A particular proportion of muscle creatine is converted to creatinine spontaneously every day. So amount of creatinine produced in the body is related to the muscle mass. The diet may influence the value of high meat intake and is freely filtered by glomerulus. It's an indicator of renal function. And the increased concentration of creatinine will be muscular dystrophies, myositis, myasthenia gravis, pregnancy and in high fever, the diabetic mellitus patients, and hyperthyroidism, and also in febrile conditions. So in all these conditions there will be a increased concentration of creatinine, we can see. And the causes of increase in serum creatinine will be, they are divided into pre-renal, renal and post-renal, as dehydration, pyloric intestinal obstruction with vomiting. As in the concentration of also it comes under the increased in urea, as renal and post-renal. Under the renal, we can see acute glomerulonephritis, malignant hypertension, chronic pyelonephritis, and the congenital cystic kidney, and hyperthyroidism and hypervitaminosis D. So post-renal, the enlargement of prostate, tumors of the bladder affecting the urate, and the post-renal will be enlargement of the prostate. So here the increased of concentration of creatinine will be, conditions are same as increased urea concentration, but in renal causes creatinine increases more than the pre-renal causes. So we can see the same causes or factors which affects the increased level of the urea and creatinine. So we'll see the estimation of creatinine as by Jaffe's method in urine and also in serum. There will be a same method. The principle includes creatinine urine reacts with the picric acid in presence of NaOH to give orange colored compound. Creatinine picrate color red at 500 nanometers. So as the column we can see blank standard in the test. We'll use 0.5 ml for the blank. We'll add distilled water and the working standard for the standard, and the diluted urine for the test. So picric acid for all the rest will be adding 10 ml, and the 0.75 normal NaOH for the all the test tubes as 1 ml. So we'll be reading the OD, and the concentration of a creatinine in 100 ml of a urine will be the formula as OD of a test minus OD of a blank. divided by the OD of standard minus OD of blank multiplied with the concentration of standard by volume of sample taken into 100 mg per dl. And here we can see the amount of creatinine excreted per day. So per day how much the amount of creatinine is excreted also can be calculated with this formula by OD of test minus OD of standard multiplied with 1500 divided by 1000, which in terms will measured in grams per day. And the creatinine estimation in the serum will be modified Jarvis method, as creatinine reacts with the alkaline picrate to produce a reddish color. Absorbance of the color is directly proportional to the creatinine concentration in the plasma, and it's measured in 500 to 520 nanometer. So here also same as in the estimation of a serum, estimation of a creatinine in the urea. So procedure will be the same. And the calculation part includes as the OD of test minus OD of blank divided by OD of standard minus OD of blank. It's multiplied with the concentration of standard by volume of a sample taken into 100 mg per dl. So the result will be the concentration of a total serum present will be represented as grams per dl. So this includes with the protein metabolism too, as the estimation of urea, creatinine and blood urea nitrogen is completed with this metabolism 2 part, and also some part of the urea. Thank you.